So I'm going to talk today just about some methods that I've tried and some of our explorations with trying to use explainable AI with neuroimaging data. So first, explainable AI has grown in interest in the neuroimaging community. So this is a review that came out a couple weeks ago from one of my former students, Farzad Farahani, who's now at Johns Hopkins. So he did this nice literature review of the different types of explainable AI methods and to which modalities they were being applied to, and kind of broke those down into what type of explainable AI method was being used. So there's certainly some decomposition-based methods, but also it seems as though model-agnostic methods are still probably the most used, just because they're so simple. We can do a pixel flip, of course. We can drop out a region of interest in the brain and just see what happens to the prediction outcome. But hopefully, we will talk about some nuances of why that may or may not provide the information that you're looking to find using these model-agnostic approaches. And then, of course, signals and gradient-based methods that have been discussed extensively this week are also increasingly being applied. So that's exciting because there seems to be a lot more growing interest, particularly since around 2017. And this is a nice word cluster association of the keywords used in the papers that we identified as explainable AI applied to neuroimaging. So for those that have developed LRP-based methods, it seems like there's growing interest in deep Taylor decomposition and layer-wise relevance propagation techniques. So exciting. So even though it's still relatively new for the neuroimaging community, there's already been some exciting new discoveries. And so one of the goals of explainable AI is, of course, can we learn something new by going through this whole exercise of training a deep learning model and then trying to see what it learned? What can humans learn from that? And so my brother's a radiologist, and he was trained, when you're trying to stage a tumor, you only look at the boundaries, the borders of the tumor. There's no information on the inside. And my brother kept thinking, yes, there's got to be something inside the tumor. But he would look and search, and to the human eye, there's really no information in there. But in fact, some deep learning models have now been trained to identify and stage tumors, and they predict very well. And in fact, there is information inside the boundaries of the tumor. So this is using texture-based features. So that's interesting. That can perhaps help human AI teams to discover how we can, work together and synthesize this information, the expert human with some of this texture-based features from explainable AI techniques. So even though explainability may be new to certain domains, it's really been a core fundament in the neuroimaging community almost since the beginning of machine learning models being applied to neuroimaging data. And one of the reasons why is because there's always been this goal, whether it was explicitly stated in the papers or just simply implied, that one would like to gain information and knowledge about the representations learned in the brain and that are used to support certain tasks and decisions, and also whether the brain is using distributed or more symbolic localized representations to understand information and to code its information. So, of course, there's tons and tons and tons of examples as to why information is probably being represented through distributed representations in the brain. But there is this famous paper, the Jennifer Aniston cell paper. I'm not sure if you're not in the neuroscience world, you may not have heard about this, but there's this very, very old idea that you might have a grandmother cell. So you might have one cell in your brain that becomes active every time you see your grandmother. And so this was actually tested here at UCLA on patients that had epilepsy, and the epilepsy was not responsive to medication, and so they had to chronically implant electrodes into the temporal lobes of patients with seizures. And they presented different celebrities. In this case, it was Jennifer Aniston. So this paper, people just call it the Jennifer Aniston cell paper. So different viewpoints of Jennifer Aniston. And sure enough, there was a cell that responded to Jennifer Aniston, whether it was a bright scene, a dark scene, a profile, or a direct image of her. And there's lots of other celebrities used in this study. I think Oprah was used and a few others, and they've also done this with moving images as well. Famous clips, Martin Luther King, I have a dream speech, different things like this have also been identified to and linked to activity in localized neurons. So that essentially provides information for both distributed and localized information coding in the brain. And so using explainable AI techniques, we may be able to move towards testing theories as to how this information is linked, how it's used, and how the brain deploys different processes to integrate this information and read it out so that it can be used for accomplishing tasks. So that's one of the main reasons why we have been very interested in decoding studies for quite a long time in the neuroimaging community. But of course, specifically with fMRI data, it is high dimensional data, so it's difficult to design decoding studies and then let alone also apply these decomposition-based techniques, et cetera, to gain insight into saliency maps. So there's something like 100,000 voxels, depending on which measurement tool that you're using. These are measured over time. The data is highly non-stationary, so the brain is plastic, and there's all these different drifts and things that occur, even over short time spans, but most certainly over days. And the measurements that we're actually measuring, it's neural activity that's effectively filtered through this temporally blurred physiologic hemodynamic response function. So there's quite a lot going on when you're trying to understand the neural code using this specific tool. But nonetheless, we have tried and explored lots of different methods, lots of different features and concepts for both encoding and decoding studies. So decoding studies, you begin with the data and try and predict the stimulus, and then encoding models work in the anti-parallel flow of information where you take the stimulus and you try to actually predict the brain activity data. And so you can perform encoding and decoding studies across different levels of granularity depending upon the features that you're interested in using. So here's a few examples, and I chose these after the conversations that I've had this week with everyone here. So here's an example with very fine granularity where you're using just the voxels and just a specific region of the brain to identify facial viewing. And so this is a nice study that used a variation on something called MVPA, multivoxel pattern analysis, with this added step of temporal embedding to help identify which aspects of the hemodynamic response function were most informative for performing this type of decoding. We've also done all kinds of studies in my group using graph-based features. So you can take the brain activity data and form these matrices using the correlations. You begin with creating a functional connectivity matrix, and then you can derive these graph measures of the brain. And the brain is interesting because it's a small world network, and you can perform learning using these graph features. So in this case, this came out just last year. We were able to make some nice predictions about sleep chronotype. So are you a night owl or a morning person? and how does that affect your productivity if you're awake at certain times of the day. So that's kind of interesting, and this was based on resting state functional MRI data. This one's also nice, which is also not at the single voxel level, but somewhere in between, these kind of derived measures using self-other resonance networks. And this was used to try and make predictions and make inferences about empathic concern. So empathy is thought to rest on this idea that we have some resonance between the self and other. So this is an interesting paper if you're interested in empathy. And I know that this group that I collaborated with is now moving on to try and think about how you would create empathic AI, if that is something that one wants to create. We've also started to use graph convolutional networks, and these do tend to work better than just your vanilla deep learning neural network for making different predictions. So in this case, you have graph representations, and you use this graph convolutional neural net that also people have talked about here. And it might not seem that exciting to you, but sometimes getting these methods adapted so that they work on this four-dimensional data is tricky. So we were excited to get this working and to make some nice predictions using the Human Connectome Project data on which task the subject was performing based on resting state activity. Wavelets also came up in some of our lunch discussions. Could wavelets be used? So yes, of course. We've used wavelets quite a lot with EEG data, and that's pretty standard for doing single trial decoding from EEG data at this point. In this specific example, we used wavelets, and we, of course, tested out multiple models, so we didn't fall into this single model fallacy, and we're able to predict autobiographical decisions. So if you're presented with, I have brown eyes, you can make predictions as to whether the person will indicate yes, they do, or no, they do not, using wavelets derived from EEG data. And then, of course, there's lots of different ways to unmix the fMRI data. So these applications kind of go back a little ways, but I think that they're very similar in spirit to some of what was discussed this week in terms of using methods to orthogonalize your data in combination with the explainable AI techniques. So in this case, PCA or ICA can be used and just applied on your input data, and then you can perform learning on those, either the spatial maps or the time series, and then make some nice autobiographical decisions. And of course, also tying in some of what was discussed earlier this week with using latent variable models to gain insight. We've applied latent variable model techniques to gain insight on multimodal data from EEG and fMRI. So you can tell what information is private versus public using things like temporal kernel canonical correlation analysis, or what information is signal versus artifact using, say, low-rank sparse decomposition. And so it is kind of amazing how well low-rank sparse decomposition works. In case anyone else is doing simultaneous EEG fMRI, it really, really works amazingly well compared to, say, Infomax or one of the vanilla ICA techniques. So lots and lots and lots of different techniques have been used to try and gain insight from these models. The simplest, of course, is just feature ranking. And there's lots of different ways to look at feature ranking in isolation or in combination. So looking at if you have hundreds of thousands of features, say voxels, performing feature ranking is very computationally burdensome, so it might make sense to do random samples and groups of features at a time. And sometimes the model itself is the interpretation. So sometimes what we're doing is exploring and testing different models to see which model best fits the data, of course. And then the winning model is then interpreted in some way as possibly representative of something happening or some aspect of brain information processing. So this is a very simple example, but I still really like this example because decision trees are so very simple, and they're just inherently interpretable. And so this is an example of using fMRI data to predict human decision making using decision trees in combination with independent component features. And it's very nice because it's simple. The latent variable technique, eliminates a lot of the covariance in the data, so makes the interpretability a lot easier. But unfortunately, there is still this problem usually where multiple decision tree architectures will perform approximately well. And so if they all perform about the same, how do we choose which model is the best one to interpret? And so if the goal is to just make some sort of output decision, it might not matter. But if the goal is to interpret the form of the function that maps the input to the output, then it is important to identify the candidate models that could produce this particular input-output mapping. So in the neuroimaging world, there's different techniques used to answer different questions. So one question might be, can we gain mechanistic insight about brain information processing by looking at the representations learned in subsequent layers of deep learning? Of course, this is an excellent way to think about deep learning and make parallels between deep learning, artificial neural networks, and biological neural networks. But again, there's sometimes this problem where multiple models will all learn something like Gabor-like features in the first layer. And so in the end, which model is the best one that best represents the brain? Model selection can be very difficult. Another question, can we generate an archetypal or prototypical example of something? So of an ADHD brain, let's say, or an autistic brain. This would be very useful for radiology students and providing archetypal examples of different diseases and different conditions. This one here, does a certain voxel or pixel or brain region contain information? So usually that's approached by pixel flipping or dropping out information in a specific region. But unfortunately, if there's redundant information, which we expect in neural code, if you drop out information, it may be rescued, so to speak, by very similar information that's redundant in the neural code. So this is not a good technique for lots of reasons. Another reason is, of course, that the deep learning models themselves perform better when you reduce the redundancy in the data. So lots of problems, I think, with the pixel flip method. In fact, techniques like drop block is used often to eliminate parts of the model, and that often boosts classification accuracy. So if you drop out a part of the brain that actually in and of itself contains information, but it's redundant, you may actually see a boost in the output. And therefore, you may conclude incorrectly that that part of the brain or those pixels did not contain information because you drop them out and then you see a boost in the classification accuracy. And then, of course, these redistribution techniques are often used to gain insight through examining the relevancy and the saliency maps. So through all these explorations, there's some challenges, I think, that keep coming up. One of them is that the noise seems to complicate fMRI and EEG data quite a lot. So the noise is, of course, mixed through some sort of unknown mixing matrix that affects the measurement channels in different ways. And noise is possibly brain rhythms. It could be related to heart rate, which should vary. There's heart rate variability is a sign of good health. We also, as I mentioned, we expect that there should be a certain amount of redundancy in the neural code. So if the neural code is, of course, dynamic because neurons cycle through this refractory state where they're quiescent. So we expect that there's probably multiple configurations of representations that should be able to convey the same information. And that can provide complications, as I mentioned, with the pixel flip type methods specifically. And also that we have this non-stationary data. And so even if we can identify core concepts that are provide evidence for a specific decision, one might expect there to be concept drift because the representations that are linked to a specific concept we know are going to evolve over time. So in this sense, there's many challenges for how we actually draw inferences and make meaningful interpretations, even when we have excellent explainable AI methods. One of them being that just because there is information contained in a specific region of the brain doesn't mean that the brain is actually using that information for that specific task. So for example, if there's one brain area that drives subsequent brain areas, regardless, the brain may or may not be using those subsequent downstream areas to accomplish a specific task. So in this sense, there could be an overinterpretation if one isn't careful about thinking deeply about the model and what the goal is of the interpretation. So here's some desirable properties for explainable AI and neuroimaging. I think that some people have addressed most of these, but I will just briefly talk about how they relate to neuroimaging. So we, of course, want our models to be sensitive in the sense that they can help us understand how expressive the model is at capturing different concepts. Completeness is a good one. Input invariance is very nice. Brought up in this Kinderman's paper that the saliency method should mirror the sensitivity of the model to input perturbations. So sometimes you can have an input perturbation, such as a mean shift, that doesn't affect the weights at all and doesn't affect the input-output predictions, but it will affect the saliency maps. And depending on which method you're using and depending on which reference points that you're using for interpretation. Of course, we want to provide the explanations in a format that the human can understand. So if you were not at the talk yesterday, I thought this was very interesting, that we shouldn't really consider deep learning models black boxes, but rather opaque boxes, because indeed we can print out all of the different weights and know everything that's happened with the learning process. But it's in this large format across so many layers that it's not very easy for the human to understand what's really happening. And then this last one here, which I think is not necessarily common, but I think in certain neuroimaging applications, it's actually crucial, which is to have at least one explainable AI technique that could be used if the goal is to provide a unified set of features or concepts or an unabridged saliency map that shows all of the features contributing to a specific decision. So, for example, going back to the brain tumor, it might be easy in the case of a brain tumor to just say, okay, run some sort of edge detection and find the borders of the tumor, and everything inside is part of the brain tumor. So that's easy. But what about seizure onset zones? So if someone's having seizures and they're not responsive, they're medication refractory, the surgeon may need to resect or remove some part of the brain. So in this case, it's very, very crucial for the patient to not just get some of the seizure onset zone, but get all of it and nothing more. So just specifically the seizure onset zone, nothing more, nothing less, just to be very both sensitive and specific. So that nice trade-off there. So what talk in this workshop would be complete without showing this image? This is probably the most famous image of this whole workshop so far. So this paper, in case you missed it somehow, is effectively showing that deep learning neural networks can sometimes learn shortcuts based on spurious correlations in the training data. So in this case, the copyright symbol was used by the classifier to make the predictions of a horse. So one might think, oh, good, my model's working. It's predicting horses so well and so accurately. But in fact, the model was using the copyright symbol. And so this is one of the big arguments for explainable AI is, of course, to make sure that your model has learned the intended concept and hasn't learned a shortcut. But I was inspired by this and thought, can we flip this the other way around? Can we introduce a copyright symbol, and what would that tell us? So one of the goals that I've been thinking about and trying to explore different ways to do sanity checks, because we need to have a way of not just finding explanations, but kicking the tires, so to speak, and making sure that the explanations themselves are not corrupted by adversarial examples that could be silent and not affect the prediction accuracy, but simply just affect the saliency maps. And so, inspired by this, we did two things. We wanted to see what noise in the wild, so to speak, how adding a specific type of noise that we know to be very common to the MRI setting, so Ricean noise is very common in the MRI setting, and see how additions of varying amounts of Ricean noise would alter the saliency maps, and find a way to quantify how those maps were altered, and to use this idea of a copyright symbol as a didactic perturbation. So to tell the model, here it is, this is the copyright symbol, but instead we used a panda, and to see how well the models did at identifying a signal that correlated very strongly with the goal of the class label. So in this case, since there were some talks on genetics, one of the kind of keys to designing a nice genetics experiment is to have a true positive and a false positive built in to help benchmark your study. And so in this case, we thought of this as being the true positive that would help us determine whether the explanation methods were indeed finding the true signal. And so we looked into different similarity ways to quantify the maps. And so we ended up using structural similarity analysis, this nice way here to quantify the distance between saliency maps before and after perturbation with Ricean noise. And unfortunately, Ricean noise does cause quite a bit of problems. In the MRI world, what often happens is you'll have a longitudinal study. Someone will come in, and then maybe the next week they'll come back. And over that time, the scanner has drifted and changed a little bit. Or even worse, you have MRI scans collected at different sites. And it turns out that data harmonization is actually a very challenging problem in the neuroimaging world. And so here's another example of just how the type of noise that we expect to find causes pretty significant differences in the saliency maps. And just testing here, two different methods, layer-wise relevance propagation and LIME, we found that, in fact, the panda was missed the vast majority of the time. So oftentimes these models would just pick one of the pandas because it was redundant information. It was the panda was introduced in the top left corner if the subject was a typically developing subject versus the right corner if the subject had ADHD. And sometimes these models identified one of the corners as being relevant but not the other, and sometimes neither one. So it is worth noting that this was done using the Investigate toolbox, and so I now know that there's newer iterations of this, and so I'm excited to try the new ones to see what we can learn. But I don't necessarily think this is a bad thing. I think the model was doing what it was supposed to, which was learn a compact set of features and concepts to make the prediction. So the model was accomplishing the task, and it was discarding the relevant information that was highly correlated. So I think, again, this is maybe not a goal of a lot of different different domains, but I think it is an important goal of neuroimaging, and possibly things like self-driving cars as well. You probably don't want a subset of a child running across the street. You probably want to know where the full child is so you don't hit any part of the child. Like, it's not okay to just hit their leg or something like this, right? So this is a nice paper discussing some of the nuances with even something as simple as linear models can actually be quite difficult to fully interpret, specifically because the models will somewhat randomly pick one of the correlated features and discard the redundant information. So I just want to end on this thought here, which is, what can explainable AI tell us about the opaque, deep, recurrent neural network inside the brain? I think that I'm very excited to know all of these new techniques and new things to try and synergize some of these techniques and use them in combination, because I do think that eventually we will get to this point where explainable AI will help tell us quite a lot about the representations learned in the human brain, and also about how they are conveyed forward to accomplish different tasks. So here's some useful references, a couple of the key papers, and some books. And yes, thank you so much.